Hello, and welcome to Sleep Archive. Tonight, we turn our gaze inward. We stare at the stars, the mountains, and the oceans, searching for the sublime in the sheer scale of the world around us. But there is a universe that exists closer to us than any of these, a universe that resides within the very eyes you are using to see and the brain you are using to listen. It is the microscopic universe, the hidden machinery of life itself. This is a world of biological clockwork, of spinning motors, winding cables, and library vaults filled with the instructions for building a human being. It is a landscape where chemistry becomes biology, where dead matter is somehow sparked into the glorious, self-replicating dance of the living. We will journey deep into the architecture of the cell, shrinking down to walk along the twisted ladder of our own DNA. We will witness the silent work of the enzymes and the structured beauty of the proteins that fold themselves into the tools of existence. But before we can touch the helix, we must first leave the world of human scale behind. We must cross the threshold of size, descending through the powers of 10 to arrive at a place where gravity means nothing and the thermal storm of atoms means everything. Prepare to make yourself very, very small. To understand the cell, we must first shatter the illusion of our normal reality. When we look at our own hands, resting quietly on the bedsheets, we see smooth skin, stable lines, and a sense of solid, continuous matter. We perceive ourselves as single, unified beings, coherent objects moving through space. But this solidity is a trick of the eye, a simplification created by our limited vision. In truth, you are not a single object. You are a galaxy. You are a colony of 37 trillion individual living entities, a vast, cooperating swarm of cells that outnumbers the stars in the Milky Way. To visit their world, we must engage in a radical act of shrinking, a mental descent into the nanoscopic. Imagine floating above your own hand like a satellite in orbit. As we begin our descent, zooming in by a factor of 10, the smooth landscape of the skin transforms. It becomes a rugged terrain of ridges and valleys, a canyon land of dead keratin scales that form the outer shield of your body. We descend further, passing through the cracks in this armor, and suddenly we are plunged into a warm, aquatic world. We have entered the interstitial fluid, the internal ocean that bathes every cell in your body. Here, the rules of physics that govern our daily lives, gravity and inertia, fade away, replaced by new, more violent forces. The first thing you notice is the motion. Down here, nothing is still. We are surrounded by the thermal storm, a chaotic, ceaseless bombardment by water molecules. Because of the warmth of the body, the water molecules are vibrating at incredible speeds, slamming into everything they touch billions of times per second. This is Brownian motion, the jittering dance of the microscopic world. If you were floating here, you would be buffeted from all sides, tossed around like a small boat in a hurricane of invisible particles. This constant agitation is the energy source that drives the machinery of life. It is the force that pushes proteins together and allows chemicals to find one another in the dark. As we stabilize ourselves in this fluid, we begin to see shapes looming out of the molecular fog. They are colossal, spherical structures, grander than any man-made space station. These are the cells. At this scale, a single cell is a leviathan, a massive, pulsating orb that dominates the horizon. It is not the simple, clear bubble you might remember from a high school textbook diagram. It is incredibly crowded. The space around us is thick with traffic. Fleets of proteins, hormones, and sugar molecules are rushing back and forth, navigating the narrow channels between the giants. It looks less like a biology lab and more like the aerial view of a futuristic megacity at rush hour, teeming with intent and purpose. The density of life here is suffocating. There is no empty space. Every cubic nanometer is packed with chemistry. We drift closer to one of these cellular giants, approaching the outer boundary. From a distance, the surface looks smooth and uniform, a continuous skin protecting the interior. But as we drift within touching distance, the surface resolves into a complex, shifting mosaic. It is alive, undulating like the surface of a sea. This is the frontier, the physical border between the chaos of the outside world and the ordered sanctuary of the living interior. It is the gatekeeper that decides what enters and what leaves. We are hovering directly in front of the cell membrane. It is not a solid wall, but a liquid crystal, a structure that manages to be both a barrier and a gateway at the same time. To enter the city, we must understand the ingenious architecture of this fluid wall, a double layer of fat that separates the living from the dead. We prepare to cross the threshold, moving from the open ocean of the body into the private universe of the cytoplasm. The barrier ahead is shimmering with a faint electrical charge, guarding the secrets within. We push forward, slipping between the molecules to enter the sanctum. As we push through the boundary, we find that the wall of the cell is not made of stone or brick, but of something far more elegant and ancient: fat. We are passing through the lipid bilayer, the universal skin of all life on Earth. Whether it is the cell of a blue whale, a redwood tree, or the bacteria on your skin, every living thing uses this exact same architectural design to separate itself from the rest of the universe. The construction of this wall relies on a simple chemical paradox involving water and oil. The building blocks here are phospholipids, strange, pin-shaped molecules with a split personality. The round head of the pin is hydrophilic. It loves water and desperately wants to be in contact with the aquatic environment. But the two long tails of the pin are hydrophobic. They hate water and are repelled by it, seeking only the company of other oils. In the chaotic, watery world of the body, these molecules spontaneously arrange themselves to satisfy both desires. They line up in two perfect parallel sheets, back to back. The water-loving heads face outwards towards the ocean of the body and inwards towards the water of the cell, shielding the water-hating tails which hide in the dry, oily middle of the sandwich. This self-assembling property means the cell membrane is self-healing. If you were to puncture it with a needle, the lipids would simply flow back together like oil on water, sealing the hole the moment the needle was removed. But as we drift through this oily layer, we notice that it is not a stagnant sheet. It is fluid, moving with the viscosity of olive oil. Floating within this sea of fat are massive, rugged shapes, proteins that look like icebergs drifting in a polar ocean. This is the fluid mosaic, a dynamic shifting surface where the machinery of the cell patrols the perimeter. These floating icebergs are the gatekeepers. If the membrane were a solid wall, the cell would starve, cut off from the fuel it needs to survive. So the membrane is studded with channels and pumps, highly sophisticated doorways that are strictly VIP only. One channel might be shaped to allow only a single potassium ion to pass. Another might be a massive pump that burns energy to shove calcium out against the tide. They act like customs officers at a busy border, constantly checking the chemical passports of every molecule that tries to enter, ensuring that nutrients get in and toxins stay out. Other proteins act as antennas, towering structures that reach out into the external void to catch passing signals, hormones like insulin or adrenaline that tell the cell what to do. They are the senses of the microscopic world, tasting the environment and relaying the news to the command center deep inside. As we emerge from the inner side of the membrane, we are finally inside. We have entered the cytoplasm. The feeling here is different. The air, or rather the fluid, is thick, viscous, and crowded with dissolved sugars and salts. We might expect the interior of the cell to be a soup, a messy bag of liquid where organelles float around randomly like vegetables in a stew. But as our eyes adjust to the dim light of the interior, we see that this is a lie. The cell is not a soup, it is a structure. Stretching out before us, disappearing into the hazy distance, is a massive, intricate lattice of cables, beams, and pillars. It looks like the girders of a skyscraper or the suspension cables of a bridge, crisscrossing in every direction to create a rigid, organized 3D space. The cell has a skeleton. It is an engineering marvel that defies the softness of the biological world, a network of dynamic scaffolding that gives the cell its shape and provides the roads upon which its cargo travels. We are not just floating in a bag of water, we are standing inside a cathedral of tension and compression. We grasp one of the thick, rope-like cables nearest to us, feeling the vibration of the city running through it. We are about to explore the bones of the beast. We transition now from the liquid skin to the solid frame, moving from the membrane to the cytoskeleton. We prepare to walk the high wires of the inner cell. If you were to poke a cell with a microscopic needle, it would not squish like a balloon filled with water. It would bounce back. It has elasticity, tension, and a defined architecture that resists the crushing pressure of the outside world. This resilience comes from the cytoskeleton, a hidden network of filaments that fills the interior of the cell like the steel girders of a massive bridge. But unlike the skeleton of a human, which is made of static calcified bone, the skeleton of a cell is a dynamic crystal. It is constantly building itself up and tearing itself down, shifting its shape to crawl, divide, or brace against an impact. As we look around the bustling interior, we identify three distinct types of architectural beams that hold this city together. The first and most delicate are the actin filaments. These are the thinnest cables, concentrated just beneath the membrane we just passed through. They form a dense, chaotic mesh, like a tangled web of spider silk glued to the underside of the skin. This mesh is the muscle of the cell. By rapidly lengthening and shortening, These filaments push against the membrane, allowing the cell to change shape, to reach out a pseudopod, a false foot, and drag itself forward. It is the mechanism of crawl, the physical force that allows a white blood cell to chase down a bacteria or a wound to close itself. Deeper in the interior, we see the intermediate filaments. These are the tension cables, thick, braided ropes that look like the steel hawsers used to anchor a suspension bridge. They are not designed for movement, they are designed for stress. They stretch across the cell from one side to the other, anchoring the organelles in place and preventing the cell from being torn apart when it is stretched or squeezed. But the true marvels of this engineering world are the microtubules. These are not ropes, they are pipes hollow, rigid tubes formed by stacking proteins in a spiral, exactly like a masonry chimney. They are the largest structures in the cytoskeleton, radiating out from the center of the cell like the spokes of a wheel. They serve two critical functions. First, they are the compression beams, the pillars that hold the cell open and prevent it from collapsing under the weight of the surrounding fluid. But secondly, and perhaps more amazingly, they are the highways. The interior of a cell is too crowded and too viscous for materials to simply float to their destination. If a protein made in the south needs to get to the north, diffusion would take far too long. So, the cell uses the microtubules as a railroad system. And walking along these rails are the most charismatic machines in the entire microscopic universe, the motor proteins. We watch in awe as a creature called kinesin steps onto the microtubule in front of us. It looks undeniably like a tiny, bipedal stick figure. It has 2 feet that physically walk along the tube, taking one step after another in a distinct hand-over-hand motion. But kinesin is not walking empty-handed. Towering above it, tethered to its tail, is a cargo container, a vesicle filled with proteins or chemical signals that is thousands of times larger than the walker itself. It is like watching a human carrying a skyscraper on their back, marching steadily along a tightrope. With every step, the kinesin burns a molecule of fuel, ATP, snapping its foot forward and binding to the track, dragging the massive load against the chaotic resistance of the cytoplasm. It is a relentless, mechanical march. If the kinesin encounters an obstacle, it pauses, waits, or sidesteps. If the road ends, it falls off. Millions of these walkers are active right now inside every cell of your body, transporting neurotransmitters down the long axons of your nerves to let you think, or dragging chromosomes apart so you can grow. They are the logistics network of life, turning the static fluid into a bustling economy of import and export. This network is organized around a central hub, a grand station from which all the major microtubule highways originate. We follow the flow of the kinesin traffic, moving deeper into the cell, guided by the converging rails. Ahead of us, looming in the center of the cytoplasm like a fortress within a city, is a structure of immense proportion. It is the largest organelle of them all, a spherical vault protected by a double-walled security perimeter. The traffic is thickening here. Thousands of motor proteins are delivering their messages to the gates, and thousands more are waiting to pick up new orders. We have reached the seat of government, the place where the master blueprint is kept safe from the chaos of the thermal storm. The highways end here. We stand before the nuclear envelope, preparing to enter the forbidden library. We transition now from the noise of the construction site to the hush of the archive. We are about to enter the nucleus. We stand before the largest and most significant structure in the cellular landscape, the nucleus. If the cell is a city, this is the Capitol building, the central library, and the fortified bunker all rolled into one. It is massive, occupying nearly 10% of the cell's total volume, a sphere so dominant that it was the first organelle ever discovered by early microscopists using primitive lenses. But up close, the first thing we notice is the security. The barrier protecting this inner sanctum is not a single wall. It is a double-layered fortification known as the nuclear envelope. Two distinct lipid bilayers, separated by a narrow gap called the perinuclear space, wrap around the contents, creating a seal that is virtually impermeable to the chaotic chemistry of the cytoplasm. This is a necessary quarantine. The information stored inside is so precious and so fragile that it cannot be allowed to float freely among the destructive enzymes and energetic motors of the outer cell. It must be kept in a clean room, isolated from the thermal storm. However, a library with no doors is useless. The information must be accessible to be read. So, the smooth surface of the envelope is punctured by thousands of massive, circular gateways known as nuclear pore complexes. These are not simple holes. They are complex, basket-like machines made of hundreds of different proteins, acting as the most sophisticated airlocks in nature. We watch as a cargo molecule approaches one of these gates. It is a transcription factor, a protein carrying an urgent message for the genes inside. It does not simply walk in, it presents a molecular ID card, a specific sequence of amino acids known as a nuclear localization signal. The pore reads this signal, and only then does the basket open. The gate dilates, creating a channel just wide enough for the protein to pass, and then snaps shut behind it, preventing any unauthorized molecules from slipping in during the exchange. It is a checkpoint of absolute strictness, ensuring that the genetic code is never touched by anything that hasn't been explicitly invited. We slip through the gate, following the messenger, and find ourselves in the nucleoplasm. The atmosphere here is different, hushed, thick, and reverent. The frenetic mechanical noise of the cytoskeleton is gone. There are no motor proteins marching here, no mitochondria burning fuel. We are suspended in a gel-like substance that holds the treasure of life. But looking around, we do not see the familiar X shapes of chromosomes that are so often depicted in textbooks. Those tightly wound structures only appear when the cell is about to divide. Most of the time, the library is open for business, and when a book is being read, it cannot be closed. Instead, the interior is filled with a chaotic, fibrous mass that looks like a bowl of transparent angel hair pasta or a tangled ball of yarn. This is chromatin. It is the genetic material in its working state, unspooled, relaxed, and spread out so that the reading machines can access the text. It floats in the nucleoplasm in great, looping territories, organizing itself into loose clouds of information. Deep in the center of this tangled cloud, we see a dark, dense region that looks like a core within the core. This is the nucleolus. It is not an organelle bounded by a membrane, but a condensate, a massive gathering of machinery that has come together to perform a single critical task, the construction of ribosomes. If the nucleus is the architect's office, the nucleolus is the factory where the builders are recruited and assembled before being shipped out to the construction site. It is the busiest part of the nucleus, a hive of activity where the fundamental tools of life are forged. But our attention is drawn back to the threads of chromatin surrounding us. This chaotic tangle is deceptive. It looks messy, but it is actually organized with a precision that defies our understanding of data storage. Somewhere in this mass of yarn is the instruction for blue eyes. Somewhere else is the recipe for insulin. Somewhere else is the code that dictated the shape of your thumb. To understand how a chemical can carry a thought, or how a molecule can store a history, we must look closer at the thread itself. We need to zoom in further, past the protein spools that hold the thread together, to see the strand in its naked form. We are descending to the atomic level to witness the most famous geometry in the history of science. We transition now from the library vault to the book itself. The tangled yarn is about to resolve into a spiral. We are standing at the foot of the twisted ladder. We have zoomed in from the city of the cell to the library of the nucleus, and finally to the tangled threads of chromatin. Now, we magnify the view one last time, stripping away the protective proteins to reveal the molecule that sits at the very foundation of biological existence. We are looking at deoxyribonucleic acid, DNA. In the textbooks of our youth, this molecule was often depicted as a flat, two-dimensional ladder, static and rigid on the page. But here, in the wet, vibrant reality of the nucleoplasm, it is nothing like a drawing. It is a living, moving sculpture, a masterpiece of architectural engineering that manages to be incredibly robust yet perfectly flexible. The shape is iconic, the double helix, but the genius of its design lies not in its beauty, but in its chemical logic. It is constructed of two distinct parts, with two distinct functions, the rails and the rungs. The rails of the ladder, the long, spiraling strands that form the outside of the helix, are the bodyguards. They are made of a repeating chain of sugar molecules, deoxyribose, and phosphate groups, welded together by ultra-strong covalent bonds. This sugar-phosphate backbone is the shield. It is negatively charged and highly stable, designed to face the harsh, watery world of the nucleus without dissolving or breaking apart. It acts as a hard casing, wrapping around the sensitive interior to protect the data stored within. But the true magic lies in the rungs of the ladder, the parts that connect the two rails together. These are the nitrogenous bases, the famous letters of the genetic code. Unlike the backbone, which loves water, these bases are hydrophobic. They hate water and want to hide from it. This chemical phobia is the force that twists the ladder. To escape the water surrounding the DNA, the bases stack themselves flat on top of each other in the very center of the molecule, piling up like coins in a roll. The two sugar backbones then twist around this stack, wrapping them in a spiral cocoon that seals out the water completely. This twisting action creates the classic helical shape. It is not a stylistic choice but a defensive maneuver, a way for the molecule to create a safe dry dock for its information in the middle of a liquid ocean. But there is a paradox here. The backbone is held together by covalent bonds, which are the strongest bonds in chemistry, hard to break, permanent and rigid. But the rungs, the connection between the two strands, are held together by hydrogen bonds. These are weak magnetic attractions, roughly 20 times weaker than the bonds of the backbone. Why would nature build its most precious archive with a spine of steel but hold it closed with a zipper of Velcro? The answer is access. If the two strands of DNA were welded together permanently, the information inside would be locked away forever, unreadable and useless. The weakness of the hydrogen bonds is the key feature. It allows the DNA to be unzipped. When the cell needs to read a gene or copy its library, a specific enzyme can slide down the helix, gently pulling the two strands apart like opening a jacket. The weak bonds snap open easily, exposing the code in the center for a split second before snapping back together the moment the machine passes. It is a perfect balance of stability and accessibility, strong enough to survive for centuries, which is why we can sequence DNA from ancient mammoth bones, but weak enough to be opened in a millisecond when life demands it. The scale of this structure is mind-bending. The DNA in a single human cell, if unraveled and stretched out in a straight line, would measure roughly 2 meters or 6 feet in length. Yet it is packed into a nucleus that is only 6 micrometers wide. This is equivalent to packing 24 miles of extremely thin thread into a tennis ball. To achieve this compression without tangling, the DNA wraps itself around spools of protein called histones, coiling and supercoiling upon itself in a fractal pattern of density. This packaging is not just for storage, it is a filing system. Genes that are not needed are wound tight, buried deep in the center of the spool where they cannot be read. Genes that are active are kept loose, dangling out in the open where the machinery can reach them. We are looking at a dynamic library that physically opens and closes its own books, shifting the shape of the helix to decide which instructions to follow and which to ignore. But what exactly are these instructions? We have admired the cover of the book and the binding of the pages, but we have not yet read the text. We must now look at the rungs themselves, the 4 simple chemicals that somehow manage to encode the complexity of an eye, a heart, or a brain. We transition now from the structure of the container to the meaning of the content. We are about to learn the alphabet of life. We have descended to the absolute bedrock of biological reality, standing upon the rungs of the twisted ladder. Here, in the atomic silence of the nucleus, we discover that the complexity of life, the difference between a blue whale, a mushroom, and a human being is not determined by different materials, but by a simple 4-letter alphabet. It is the ultimate reductionist marvel. The entire diversity of the biosphere is written using only 4 chemicals, 4 nitrogenous bases that form the steps of the helix: adenine, thymine, cytosine, and guanine. We know them simply as A, T, C, and G. These molecules are not magical. They are simple carbon-nitrogen rings shaped like distinct puzzle pieces with specific geometries. And they follow a single, unbreakable rule of attraction known as complementary pairing. Because of their shape and the alignment of their hydrogen bonds, adenine can only hold hands with thymine. They are a perfect molecular match, locking together with 2 hydrogen bonds like a plug fitting into a socket. Similarly, cytosine can only pair with guanine, locking together with 3 hydrogen bonds. An A will never bond with a G, a T will never bond with a C. This strict exclusivity is the secret to life's ability to copy itself. If you have one strand of DNA that reads ACTG, you instantly know with 100% certainty that the opposite strand must read TGAC. The information is redundant, mirrored on both sides of the helix, meaning that if one side is damaged, the cell can easily repair it by looking at the partner strand to see what is missing. But the true genius of the system is not in the chemistry of the bases themselves, it is in their sequence. Just as the letters E, A, and T mean nothing on their own, but can be arranged to form EAT or TA or 8A, the bases of DNA acquire meaning only through their order. A sequence of 3 bases forms a codon, a genetic word. For example, the triplet GAG is the code for glutamic acid. Change just one letter, turn that last G into a T to make GAT, and the word changes to aspartic acid. String these 3-letter words together into a sentence that is thousands of letters long, and you have a gene, a complete instruction manual for building a specific protein. This is a quaternary code. Our computers run on binary code using 2 states, 0 and 1. Life runs on quaternary code using 4 states, A, C, T, and G. This makes DNA an incredibly dense storage medium. A single gram of DNA can theoretically store 215 petabytes of data, enough to hold all the movies ever made, with room to spare. The sheer length of the code inside you is staggering. The human genome consists of roughly 3 billion base pairs. If you were to read this code out loud, reciting 1 letter per second, without stopping for sleep or food, it would take you nearly 100 years to finish the book of yourself. And yet, despite this immense length, the difference between you and the person sitting next to you is tiny. Human beings share 99.9% of this sequence identical to one another. All the variation we see in humans' height, eye color, susceptibility to disease, talent is contained in that tiny 0.1% of spelling variations. Furthermore, we share 98% of our code with chimpanzees, 85% with mice, and even 50% with bananas. This reveals the deep, common ancestry of all things. The code was written once, billions of years ago, and has simply been edited, shuffled, and rearranged ever since. There is no new DNA language. There is only the ancient dialect of A, C, T, and G. However, a library filled with books is useless if the books never leave the shelf. DNA is a passive molecule. It sits in the safety of the nucleus, stable and unreactive. It does not build anything. It does not fight bacteria. It does not digest food. It is merely the blueprint, not the carpenter. For the instructions stored in the helix to actually do something, they must be copied and taken out of the vault to the construction site. The master copy must never leave the safety of the nucleus. It is too precious to risk damage in the chaotic cytoplasm. So, the cell faces a logistical problem: how to get the information out without moving the source. The solution is to create a disposable photocopy. We are about to witness the birth of a messenger. We transition now from the static code to the active transcription. The helix is about to be opened. The library is quiet, but it is not dormant. Deep within the tangled clouds of chromatin, a massive machine is hunting. It is an enzyme, a complex of protein subunits roughly the size of a small mountain compared to the atoms it manipulates. This is RNA polymerase, the great scribe of the cell. Its mission is singular and critical: to locate a specific gene, perhaps the instruction for creating a molecule of collagen to repair a scratch on your skin and make a perfect, portable copy of it without damaging the original. It glides along the DNA strand like a monorail train, scanning the chemical terrain for a specific sequence of letters known as a promoter. This is the start here sign, a genetic beacon that marks the beginning of a gene. When the polymerase finds this landing site, it clamps down with a shudder of chemical bonding, anchoring itself firmly to the helix. Then the violence begins. The enzyme acts as a plow. It pushes forward, physically wedging itself between the two strands of the DNA ladder. With the force of a molecular icebreaker, it shatters the weak hydrogen bonds holding the A's and T's together, prying the helix open to create a small bubble of exposed code. The DNA is now vulnerable, its secret sequence laid bare in the heart of the machine. But the polymerase does not pause. It reaches out into the surrounding nucleoplasm, snatching up free-floating nucleotides, the raw letters of the alphabet that are drifting by in the thermal storm. It grabs these letters and slams them into place against the exposed DNA strand, matching them one by one. If the DNA reads a C, the machine grabs a G. If the DNA reads a G, the machine grabs a C. But here, there is a subtle shift in the language. If the DNA reads an A, the machine does not grab a T. Instead, it grabs a U, uracil. This is the hallmark of ribonucleic acid, or RNA. Uracil is a slightly different molecule than thymine, cheaper for the cell to produce and less stable. This instability is intentional. Whereas DNA is written in permanent ink to last a lifetime, RNA is written in pencil. It is designed to fade, to be used once or twice and then discarded. As the polymerase races down the gene at a speed of roughly 50 letters per second, a long, single strand of mRNA, messenger RNA, begins to trail out behind it like a growing tail. This is the transcript, the physical photocopy of the instructions. The process is rapid and relentless. Behind the machine, the DNA strands instantly snap back together, the helix sealing itself shut as if nothing had happened, preserving the master copy in its pristine state. But the copy is not yet ready to leave the vault. The raw transcript is messy. It contains introns, junk sequences of gibberish that break up the actual instructions. Before the message can be sent, a team of editing machines called spliceosomes descends upon the RNA strand. They act like film editors, cutting out the junk scenes and pasting the meaningful exons together to create a seamless, coherent narrative. They cap the front of the strand with a chemical helmet to protect it from attack, and they add a long tail of adenine bases to the end, a poly-A tail, which acts like a burning fuse. The longer the tail, the longer the message will survive in the cytoplasm before it is eaten by enzymes. It is a built-in self-destruct timer. Now, the message is complete. It is a sleek, single-stranded ribbon of code carrying the exact recipe for a protein. It drifts away from the gene, floating through the nucleoplasm toward the edge of the world. It approaches the massive basket of the nuclear pore complex we passed earlier. This time, the gate recognizes the export signal stamped onto the RNA. The basket opens, and the ribbon snakes its way out, leaving the safety of the library behind. It emerges into the cytoplasm, the chaotic, bustling city of the cell. It is instantly buffeted by the thermal storm, but it does not wander aimlessly. It is being hunted. Floating in the cytoplasm are thousands of two-part machines, dark and enigmatic, waiting for a message to read. They are the factories of the cell, the only structures capable of turning the abstract information of the code into the physical reality of matter. The ribbon of RNA drifts toward one of these giants, and with a click of molecular recognition, it is captured. The translation is about to begin. We transition now from the scribe to the builder. We are entering the universal factory. The machine that captures the floating ribbon of RNA is the ribosome. It is, without exaggeration, the most important machine in the history of life. Every living thing, from the bacteria in a deep-sea vent to the cells of your own brain, uses this exact same device to build itself. It is the universal constructor. Up close, the ribosome looks like a giant molecular mushroom composed of two separate pieces, a small subunit that acts as the platform and a large subunit that acts as the machinery. They float separately in the cytoplasm, dormant and silent, until they detect the arrival of a messenger RNA. Instantly, they snap together, clamping the mRNA strand between them like a piece of tape in an old cassette player. The machine hums to life. Its job is translation, to translate the language of genetics into the language of proteins. But the ribosome cannot create the materials, it is only the assembler. It needs a delivery service. Enter the tRNA, or transfer RNA. These are small, L-shaped molecules that dart around the cytoplasm like frantic delivery drones. On one end of the L shape, they carry a specific three-letter code, the anticodon that matches the code on the mRNA. On the other end, carrying it like a backpack, is a single amino acid, a physical building block of matter. The process is a blur of mechanical precision. The ribosome reads the first three letters of the mRNA tape. Let's say it reads AUG. It waits. Out of the chaotic swarm of the cytoplasm, a tRNA with the matching code UAC slots into place, clicking into the machine. It carries the amino acid methionine. The ribosome accepts the delivery, holding the methionine in place. Then, it ratchets forward one step, pulling the tape through to reveal the next three letters. Another tRNA flies in, matching this new code, carrying a different amino acid, perhaps leucine. Now, the magic happens. The ribosome uses its internal energy to forge a chemical bond between the methionine and the leucine, welding them together. It kicks the empty tRNA out the back door to go reload and moves the tape forward again. Click, weld, move. Click, weld, move. It is a relentless assembly line, processing roughly 20 amino acids per second. Out of the top of the ribosome, a long, growing chain of beads begins to emerge. This is the polypeptide chain. It is not yet a functional machine. It is just a string of raw materials, a linear sequence of chemicals. But the sequence is specific. If the gene was for hemoglobin, the beads are arranged in the exact order needed to catch oxygen. If the gene was for keratin, the beads are arranged to form hair. As the ribosome reaches the end of the tape, it hits a stop codon. This is the termination signal. A release factor enters the machine, severing the bond and releasing the newly formed chain into the wild cytoplasm. The ribosome splits apart, its job done, ready to find the next message. But the chain it released is useless. It is a floppy, wet noodle of chemicals, drifting aimlessly in the water. For this string of beads to become a tool, to become a lock, a pump, or a motor, it must do something extraordinary. It must change its shape. It must transform from a 2D line into a 3D object. We watch as the string begins to twitch, guided by the invisible laws of physics. It is about to perform the most complex origami in the universe. We transition now from the assembly line to the sculpture. We are about to witness the art of folding. The newly minted protein floats in the cytoplasm, a long, shivering chain of amino acids. At this moment, it is chemically complete, but functionally useless. It is like a flat piece of metal that has not yet been stamped into the shape of a key. It has the material, but not the geometry. And in the microscopic universe, geometry is everything. Shape determines function. If the protein is shaped like a cup, it will catch signals. If it is shaped like a hook, it will pull chromosomes. If it is shaped like a tunnel, it will allow water to pass. So, the chain must fold. This process happens at blinding speed, often in microseconds, driven by the same fundamental laws of attraction and repulsion that built the cell membrane. The primary driver is hydrophobicity. Just like the oil in the membrane, certain amino acids in the chain hate water. In a desperate attempt to escape the wet cytoplasm, they collapse inward, burying themselves in the center of the tangle to create a dry core. Simultaneously, the water-loving amino acids rush to the outside, forming a protective shell. This sudden violent collapse is the first step, scrunching the long line into a rough globule. Then, the fine-tuning begins. Positive charges on the chain seek out negative charges, snapping together like magnetic buttons to lock loops in place. Sulfur atoms on distant parts of the chain find each other and forge disulfide bridges, strong covalent staples that pin the folds together permanently. Sections of the chain coil up into elegant spirals known as alpha helices, or lay flat in pleated sheets called beta sheets. It is a chaotic dance of atomic forces, a self-assembling puzzle that solves itself. But the stakes are incredibly high. If the protein folds incorrectly, if a loop twists left instead of right, it becomes denatured. A misfolded protein is not just useless, it is dangerous. It can form sticky clumps called aggregates that gum up the machinery of the cell, leading to disasters like Alzheimer's or Parkinson's disease. To prevent this, the cell employs a dedicated team of disaster relief workers. These are the chaperones. These massive, barrel-shaped proteins wander the cytoplasm looking for struggling chains. When they find a protein that is having trouble folding, they capture it. They swallow the chain whole, pulling it inside their hollow interior. Inside the belly of the chaperone, the environment is isolated. There is no water, no crowding, and no interference from other molecules. It is a private dressing room. Safe inside, the protein is given the time and space to untangle itself and fold correctly. Once the shape is perfect, the chaperone opens its lid and ejects the finished product. The transformation is complete. The floppy string of beads has become a rigid, three-dimensional machine. It looks alien, a lump of bumpy, twisted matter with crevices, pockets, and arms. But every bump and every crevice is there for a reason. One specific pocket on its surface is shaped exactly down to the atom to fit a specific molecule in the cell. This is the active site. The protein is now an enzyme, a tool ready to perform a specific task. It is a lock waiting for a key. And when that key arrives, the protein will not just sit there, it will move. We transition now from the passive structure to the active work. We are about to witness the spark of chemistry. The cell is full of finished proteins, floating in the cytoplasm like tools in a vast, gravity-free workshop. But tools do not work by themselves. A hammer lying on a table cannot drive a nail. In the macroscopic world, tools need a human hand to guide them. In the microscopic world, the tool is the hand. These are the enzymes, the tireless, unthinking workers that perform every single chemical reaction that keeps you alive. To understand why they are necessary, we must understand the stubbornness of matter. Most chemical reactions are slow. If you were to leave a piece of bread on a counter, it would eventually break down into simple sugars and energy, but it would take years of slow oxidation. Life does not have years, it needs the energy now, in milliseconds. This is the enzyme's job, to make the impossible easy. We watch one of these workers, a massive, globular protein called hexokinase, drift through the fluid. It has a deep, specifically shaped groove carved into its side, a pocket that looks like a mouth. It is hunting for a specific molecule, glucose. A sugar molecule drifts by, buffeted by the thermal storm. It hits the enzyme. If it hits the wrong spot, it bounces off harmlessly. But if it hits the groove, something magical happens. The shape of the sugar matches the shape of the groove perfectly. It slides in, and with a satisfying click, it docks. This is the lock and key mechanism, but it is not a passive fit. The moment the sugar docks, the enzyme moves. It clamps down, wrapping its atomic jaws around the sugar in a process called induced fit. It is like a molecular hug. By squeezing the sugar, the enzyme physically bends the molecule, putting stress on its internal bonds. It twists the sugar just enough to weaken the connection between its atoms, making it vulnerable. Then, with a sudden release of energy, the bond snaps. The enzyme opens its jaws and releases the broken pieces, the product, back into the cytoplasm. The worker instantly resets, its mouth open again, ready for the next victim. Hexokinase can perform this action thousands of times per second. Other enzymes are even faster. Carbonic anhydrase, which clears carbon dioxide from your blood, can process 600,000 molecules per second. Without these machines, the chemistry of life would grind to a halt. Your digestion would stop, your muscles would freeze, and your thoughts would cease because the reactions would simply take too long to happen. But this speed comes at a cost. Every time an enzyme snaps a bond or builds a new one, it often requires an investment. It needs a spark to start the engine. It needs currency. The hexokinase we just watched didn't just break the sugar for fun, it used a molecule of energy to do it. This currency is a small, volatile molecule called ATP. It is the electricity of the cell, the universal battery that powers the motors, the pumps, and the enzymes. But where does the battery come from? To find the source of this power, we must look for the power plant. We turn our attention to a large, bean-shaped organelle drifting nearby, a structure that looks strangely different from everything else in the cell. It has a double skin, and inside, it holds a secret that predates the existence of animals. We are about to enter the furnace. We transition now from the worker to the generator. We are entering the ancient engine. We approach the mitochondria. It is the second largest organelle in the cell and easily the most recognizable. Shaped like a kidney bean, it floats in the cytoplasm with a sense of autonomy, moving slightly independently of the cytoskeleton's traffic. But as we cut through its outer skin, we discover that its architecture is radically different from the rest of the cell. It has two membranes. The outer membrane is smooth, a simple skin that acts as a container. But the inner membrane is wrinkled, folded back and forth on itself like a piece of fabric stuffed into a small box. These deep folds are called cristae, and they are the secret to the engine's power. By folding the membrane, the mitochondria increases its surface area 1,000-fold, creating acres of space to pack in the machinery of energy production. Studding these folds are millions of tiny mushroom-shaped turbines called ATP synthase. This is the end of the line for the food you eat. When you digest a meal, the sugars and fats are broken down and shipped here to the matrix, the deepest, innermost room of the mitochondria. Here, a violent chemical reaction takes place. The mitochondria strips the electrons from the food and feeds them into a chain of pumps in the membrane. These pumps push protons out of the matrix, creating a massive pressure dam, a battery charged with potential energy. Then, the dam breaks. The protons rush back in through the turbines, spinning them at thousands of revolutions per minute. This mechanical rotation smashes a phosphate group onto a spent molecule, recharging it into ATP. It is a hydroelectric dam on a molecular scale, generating the currency that pays for every thought, every heartbeat, and every movement you make. But there is something strange about this power plant. If we look closely at the matrix, floating amidst the turbines, we see something that shouldn't be there. It is a loop of DNA, but it is not the linear, complex DNA we saw in the nucleus. It is a simple, circular ring, exactly like the DNA found in bacteria. Furthermore, the mitochondria has its own ribosomes, its own timetable for reproduction, and a double membrane that looks suspiciously like the skin of a gram-negative bacteria. This is the smoking gun of the endosymbiotic theory. Billions of years ago, the mitochondria was not an organelle. It was a free-living bacteria, a tiny predator that swam in the ancient oceans, hunting for food. One day, it was swallowed by a larger, more primitive cell. But instead of being digested, something miraculous happened. The little bacteria survived. It struck a deal with its captor. I will generate energy for you if you provide me with food and shelter. The partnership was so successful that it became permanent. Over eons, the little bacteria lost its ability to live alone, becoming a captive power plant, passed down from mother to child for generations. Every single mitochondria in your body today is a descendant of that original, swallowed guest. You are not just one organism, you are a colony. You are a hybrid creature, powered by an alien engine living inside your cells. With the energy pouring out of these turbines, the cell can now afford to do something more than just survive. It can manufacture. The proteins we saw being folded earlier need to be sorted, packaged, and shipped to their final destinations. We need a logistics center. We leave the hum of the power plant and head toward a massive, folded maze that surrounds the nucleus. We are entering the shipping lanes. We transition now from the engine to the warehouse. We are about to enter the Golgi. We leave the ancient furnaces of the mitochondria and drift back toward the center of the city, toward the massive structure that hugs the nucleus. This is the endoplasmic reticulum, or the ER. If the nucleus is the library, the ER is the factory floor that surrounds it, a sprawling, labyrinthine network of flattened sacs and tunnels that accounts for more than half of the total membrane in the cell. It is so large that it is folded over and over on itself, looking like a stack of deflated balloons pressed together. We approach the section known as the rough ER. It gets its name from the texture of its surface. It is studded with millions of ribosomes, the tiny factories we met earlier. But these ribosomes are not floating free. They are docked permanently onto the skin of the ER, injecting their newly made protein chains directly into the interior of the tunnels. This is the beginning of the secretory pathway. The proteins made here are not meant for the cytoplasm. They are meant for export. They are the hormones that will travel to your brain, the antibodies that will fight a virus in your blood, or the collagen that will build your bones. Inside the dark, fluid-filled tunnels of the ER, the proteins are folded and checked for quality. If they pass inspection, they are packaged for travel. A section of the ER membrane begins to bulge outward, wrapping around the protein cargo. It pinches off, creating a small spherical bubble called a transport vesicle. This bubble is a molecular shipping container. It is grabbed by a motor protein, our friend kinesin, and dragged along the microtubule tracks toward the next destination, the Golgi apparatus. The Golgi is the cell's post office. It looks distinct from the ER, a stack of smooth, disconnected saucers floating near the edge of the cell. The vesicle arrives at the receiving dock, the cis face of the Golgi, and fuses with it, dumping its cargo inside. Here, the proteins undergo their final processing. As they move through the stack, from one saucer to the next, enzymes modify them. They attach carbohydrate tags, sugar molecules that act as barcodes or shipping labels. These tags tell the cell exactly where the protein needs to go. Does it belong in the lysosome to digest waste? Does it belong in the membrane to act as a pump? Or does it belong outside the cell entirely? In the final chamber of the Golgi, the transface, the sorting happens. proteins with the same shipping label are gathered together and packed into a final, massive delivery vesicle. We follow one of these vesicles destined for the outside. It is a large bubble filled with thousands of insulin molecules. It travels all the way to the edge of the world, to the great lipid wall of the cell membrane. When it touches the wall, a remarkable fusion occurs. The skin of the bubble merges seamlessly with the skin of the cell. The bubble opens up to the exterior, turning inside out and vomiting its contents into the bloodstream. This is exocytosis. It is the moment the internal work of the cell becomes the external reality of the body. The insulin drifts away to lower your blood sugar, a product of the complex assembly line we just witnessed. The cell is a master of logistics, a machine of perfect input and output. But this machine has a shelf life. Proteins wear out, membranes degrade, and DNA accumulates damage. The cell cannot live forever. To ensure the survival of the line, it must perform the most dangerous and complex stunt in biology. It must clone itself. It must tear its own body in half to create two new lives from one. The factory is shutting down. The library is about to be packed up. We transition now from the daily grind of survival to the miracle of division. We are about to witness the dance of chromosomes. The hum of the cellular city changes. The constant traffic of the motor proteins slows down, the manufacturing in the ER halts, and the daily business of metabolism is put on hold. The cell has received the signal to divide. It is entering mitosis, the most dramatic event in the life of a cell, where the entire architectural structure we have explored is dismantled and rebuilt in a matter of hours. The first sign of this transformation happens in the nucleus. The loose, tangled clouds of chromatin, the open books of the library, begin to tighten. They wind themselves around the histone spools, twisting tighter and tighter, condensing until they form the thick, solid structures we recognize. The X-shaped chromosomes appear. This condensation is critical. Trying to pull the loose strands of DNA apart would be like trying to separate two bowls of spaghetti. They would tangle and snap. By packing them into rigid bars, the cell makes them movable. Simultaneously, the nuclear envelope, the double-walled fortress that has protected the DNA for the cell's entire life, begins to dissolve. It breaks apart into tiny vesicles, leaving the chromosomes exposed in the open cytoplasm. Now, the architect takes over. Two small structures called centrioles move to opposite ends of the cell, like the north and south poles of a globe. From these poles, they begin to shoot out thousands of microtubule cables, creating a massive, football-shaped cage known as the spindle apparatus. These cables are fishing lines. They grow rapidly toward the center of the cell, searching for the chromosomes. When a cable hits the center of an X chromosome, it latches on. A tug of war begins. The cables from the North Pole pull one way, the cables from the South Pole pull the other. This tension drags all the chromosomes to the dead center of the cell, lining them up in a perfect, single-file row known as the metaphase plate. It is a moment of total stillness, a pause for inspection. The cell checks the tension. Is every chromosome holding hands with a cable from both sides? If the answer is yes, the trigger is pulled. the glue holding the center of the X together dissolves. Suddenly, the chromosomes snap apart. The motor proteins on the spindle fibers roar to life, dragging the separated halves toward the opposite poles. They move at a speed of 1 micrometer per minute, slow to us, but a sprint for the molecular world. The single library has become 2 identical archives, safely relocated to opposite ends of the room. Now, the cell must physically split. A ring of actin filaments, the same muscle fibers that allowed the cell to crawl, forms around the equator of the cell, just beneath the membrane. It acts like a drawstring on a bag. It begins to tighten, cinching the waist of the cell inward. The membrane pinches down, deeper and deeper, until the 2 sides touch. With a final pop of membrane fusion, the single giant becomes 2 smaller, independent cells. The nuclear envelopes reform, the DNA relaxes back into its loose cloud, and the machinery of life hums back on. This process has happened trillions of times to make you who you are. But where did this choreography come from? This complex dance of spindles and motors is too sophisticated to have been the first step. To understand the true nature of the cell, we must look back, way back, before animals, before plants, and before the nucleus itself. We must go to the beginning. We transition now from the complexity of the modern cell to the mystery of its origin. We are entering the primordial spark. To see the origin of the machine, we must rewind the clock 4 billion years. We must strip away the blue sky, the green forests, and the oxygen in the air. We arrive at the Hadean Earth, a planet that looks more like a wound than a home. The oceans are green with iron, the sky is red with carbon dioxide, and the surface is battered by constant meteorite impacts and volcanic eruptions. It is a sterile, violent world. But in the quiet, heated vents of the deep ocean, or perhaps in the drying clay pools of a volcanic island, a new kind of chemistry is beginning to stir. This is the moment of abiogenesis, the genesis of life from non-living matter. It likely started not with DNA, but with RNA. In this RNA world, random chemical combinations created chains of nucleotides that had a unique ability. They could fold up and act like enzymes, but they also carried information. One day, by pure chance, a specific chain formed that could do something miraculous. It could copy itself. It was a clumsy, slow replicator, prone to errors, but it was persistent. It began to consume the organic molecules around it, making more and more copies of its own sequence. Darwinian evolution had officially begun. The molecules that copied themselves faster and more accurately survived, while the others fell apart. But these naked replicators were vulnerable. They were exposed to the harsh acids and shifting currents of the open ocean. They needed a shelter. They found it in the form of the lipid bubble. Fatty acids, created by the heat of the vents, spontaneously formed microscopic spheres in the water primitive versions of the cell membrane we studied earlier. By chance, one of these self-replicating RNA strands became trapped inside a lipid bubble. This was the game changer. Inside the bubble, the concentration of chemicals remained high. The environment was stable, protected from the dilution of the sea. The protocell was born. Over hundreds of millions of years, this primitive bubble evolved. It swapped the unstable RNA for the durable DNA vault. It developed protein pumps to control its internal chemistry. It enslaved the energy of the proton gradient. Eventually, out of this chaotic experimentation, a single survivor emerged. We call it Luca, the last universal common ancestor. It lived roughly 3.8 billion years ago. It was a simple, single-celled organism, likely living in a hydrothermal vent, feeding on hydrogen gas. It was not much to look at, but it possessed the master key. Every single living thing on Earth today, from the bacteria on your phone screen to the blue whale in the ocean, from the mushroom in the forest to humans, is a direct, unbroken descendant of that one cell. Luca is the grandmother of us all. The machinery we have explored tonight, the ribosome, the ATP synthase, the genetic code, was perfected by Luca and passed down, copy by copy, for 4 billion years. The flame has never gone out. Through ice ages, asteroid impacts, and mass extinctions, the cell kept dividing. It is a continuous chain of life that connects your hand to the stone of the ancient Earth. You are not just living on the planet, you are a piece of the planet that has learned to remember. But as we look at this chain, we realize that the distinction between the living cell and the dead universe is thinner than we think. The atoms in the DNA are the same atoms in the rock. The energy in the mitochondria is the same energy in the lightning. We are ready to zoom out one last time. We transition now from the origin of life to the unity of matter. We are entering the fade. We have walked the highways of the cytoskeleton, listened to the hum of the ribosomes, and stood in the silent vault of the nucleus. We have seen the city of the cell for what it truly is, a miracle of engineering, a place where the chaotic laws of chemistry are tamed into the orderly dance of life. But as we prepare to leave this microscopic universe, stepping back out through the fluid wall of the membrane, we are left with one final, haunting realization. There is no magic ingredient. If you were to take a cell apart, atom by atom, and place the pieces in a pile, you would find nothing but common dust. You would find carbon, the soot of a dying star. You would find hydrogen, the ash of the Big Bang. You would find oxygen, nitrogen, and phosphorus, simple, dead elements that can be found in any rock or cloud of gas in the galaxy. The cell contains no life force, no special energy that is absent from a stone. The difference between you and the chair you are sitting on is not what you are made of, but how you are arranged. Life is not a substance, it is a pattern. It is a momentary eddy in the flow of entropy, a brief flash of order that borrows energy from the sun to build a cathedral out of dust. And this pattern is ancient. The energy that is beating your heart right now is the same energy that powered the first bacteria in the Hadean Ocean. The code that is written in your DNA is the same language that was written in the first protocell. We are not separate from the universe. We are the universe looking at itself. We are the way the cosmos has learned to think, to feel, and to wonder. As we zoom out, the massive sphere of the cell shrinks away. It becomes a speck, then a point of light, then nothing. We return to the scale of the human body, lying quietly in the dark. But we perceive it differently now. We know that beneath the stillness of the skin, there is a frantic, silent industry. Trillions of motors are walking. Trillions of pumps are spinning. Trillions of libraries are opening and closing. And they do it all without you having to ask. They do it while you work, while you dream, and while you sleep. The machinery of life is vigilant so that you can rest. The thermal storm quiets down. The molecular vibrations slow to a gentle hum. The heavy doors of the archive close. The twisted ladder hangs motionless in the dark. We drift in the knowledge that we are built of stardust, held together by the fragile, beautiful laws of chemistry. We are a galaxy within a galaxy. And now, the galaxy sleeps. The work is done.